stem cells through mature B cells. And we will certainly see sort of thematic similarities between what happened to T cells in the thymus and B cells here in the bone marrow. Although, of course, some of the details will be different because we're looking at the two different lineages. So this should take us today and Friday. Next Monday and Wednesday we'll do chapter 11, which goes back to T cells and looks at their activation, differentiation, and memory. And then we'll do the same thing for B cells in chapter 12 after that. So we should get chapter 11 done probably Monday and Wednesday of next week, and that will be the perfect cutoff for the third exam, which is the following Wednesday the 16th. So that should cover then 8, 9, 10, and 11 on the next exam. And then for the fourth exam, we'll do 12, 13, probably 15 on allergies and 17 on vaccines. I think will probably be all the time we have left. Okay, so we'll get those chapters in for the final, I think. We'll see what happens. All right, so B cells. Okay, so this is sort of the overview figure that gives us some of the major things that we're going to see sort of happening. So of course, for most organisms, B cells are going to be made and mature in the bone marrow, although originally, and that's helpful, B for bone marrow, like T for thymus, but of course originally this work was done in birds, and for birds, actually the B is not really bone marrow, the B is the bursa of fabricius, whatever the hell that is. Birds, but we're not going to pay attention to birds. We'll do B for bone marrow, but technically that's what the B stands for. And we will start out in the bone endosteum with hematopoietic stem cells, and as we'll see, they will progress through a series of divisions that make them more and more committed to eventually and exclusively the B cell lineage. We saw something similar happening with the T cells. So we'll focus on them through the common lymphoid progenitor, which we talked about with the T cells that can give rise to either B or T cells. But since these CLPs will stay in the bone marrow, they'll become B cells. And then we'll watch them go through these stages. Actually, there's one before this. There's the pre-pro B cell stage, then the pro stage, then the pre-stage, then the immature stage. So we'll define all of these different stages that they progress through, again, mostly based on cell surface markers on the outside and stuff that's going on like recombination steps on the inside, essentially. So eventually we'll get the B cell receptor going, and we'll get to the immature B cell stage where we will see negative selection occurring. We don't need any positive selection for B cell receptors because we never ever want them doing anything with self, right? Unlike T cell receptors, we needed them to recognize self-MHC at least, so there had to be that positive selection. Here with B cells, we just negatively select anything that has any remote possibility of attaching to self peptides. So there's really just negative selection to talk about to remove self-reactive cells. And then that will happen presumably partly in the bone marrow, but perhaps also continuing in the secondary tissues. So immature ones that have been selected for will leave the bone marrow and then go to secondary organs like the spleen, where we'll go through a couple of more phases called the transitional 1 and transitional 2 phases before they become fully mature B cells ready to be activated, ready to make antibody. So we'll go through all of these steps as we go through this. Okay. Just a little tiny bit about the site of hematopoiesis. We kind of just automatically think it's always the bone marrow, and it certainly is for adults. However, embryonically, it goes through a sort of a series of places where various pieces of hematopoiesis take place. It actually starts in the yolk sac embryonically, where the first rough equivalents to red blood cells are made. The first cells that we would really call hematopoietic stem cells will appear in this section of the embryo called, well, abbreviated the AGM for aortic gonad mesonephros region. Yes, presumably because that's what derives from that section of the embryo. So that's where we first see the sort of rough equivalent of what we would call hematopoietic stem cells. Eventually, they will start seeding other portions, including the yolk sac. We'll see the movement of the yolk sac, also the placenta, in the red section here, and the fetal liver, that section there. So they'll kind of move to those three places and start generating a variety of blood cells. Okay, so we'll move from the yolk sac to the AGM, to then the fetal liver, the placenta. Eventually, near birth, all of this then shifts to the bone marrow where it continues after birth. Okay, but there are multiple sites of hematopoiesis embryonically, okay, before it all shifts to the bone marrow. So just to point that out there. Okay. Well, actually, this is kind of as bad as it looks. The main point of this picture, though, is to demonstrate the importance of stromal cells. Okay, we've talked about them here and there. We've talked about them as the main arbiters of selection in the thymus. We'll have the same kind of idea here in the bone marrow. Interestingly, they don't tell us what's the derivation of stromal cells until chapter 10. Interestingly, it comes from the Greek for mattress, I guess because they are supporting cells like a mattress is. Okay, so these stromal cells, as you can see just from the sort of little surface circles here with different colors, will be expressing different cell surface markers to which these maturing B cells will be able to attach. Okay, so adhesion molecules will play a role in order to keep the B cell at specific stages of its development in different sort of micro niches within the bone marrow so that they're getting the right cocktail of cytokines in order to mature, from pre-pro to pro, pre-pro to pre to pro, etc. Okay, so that's really what this is really trying to show, is that as it's sort of handed off from one type of stromal cell to another in a slightly different place in the bone marrow, it's going to get different signals that will allow it to kind of continue its maturation. That's really the key idea from this whole complicated looking picture. But as you can see, they will be moving from, again, closer to the medullary vascular to into the ostium and things like that. So things do move around within the bone marrow. That's kind of really what that's showing. Okay. This slide is really taking us through the early portion or the early steps in differentiation. And once again, similar to what we looked at in the thymus, these are all identified by different cell surface markers, well, mostly cell surface markers, although RAG and TDT, of course, are in the nucleus that will allow for recombination of the immunoglobulin genes. So you can see that those turn on a little bit later in the process. So if we start out with our classic hematopoietic stem cell, that can become anything, right? So that in the bone marrow is expressing those two cell surface markers, c-KIT and SCA1, which are the sort of, at least among the classic things you're looking for to sort of isolate hematopoietic stem cells. But at this point, since this can become basically anything, these cells are often described, especially in terms of B and T cells, as lin minus, which lin means lineage. So they're lineage minus because they're not expressing any specific proteins for a particular lineage, which we wouldn't expect from a hematopoietic stem cell, right? They can become anything. So at that point, they shouldn't be saying, oh, I'm going to be a B cell, because that's not it's not the time for that yet. But well, remember how hematopoietic stem cells are thought to work, right? They have an asymmetric cell division, right? So one of the cells remains a hematopoietic stem cell, the other then starts to differentiate. So the next stage in the development are called multipotent progenitors, MPPs, and we can distinguish them because they start expressing CD34 on their surface, and they're going to be one step sort of removed. So these are going to have a slightly more limited possibility, well, especially for self-renewal, okay? Not necessarily for becoming all the different cells, but they certainly aren't able to self-renew like the hematopoietic stem cell. So that's one characteristic that they sort of lose, one step removed from them. But they can still become basically all the major cell types, again, which is what we would expect. As it gets more signals, some of these cells will then progress, again, as we're moving towards B cells, through the lymphoid primed multipotent progenitor cell, LMPP stage. And that we see is characterized by turning off CD34 and turning on FLT7, FLT3, FLT3, which in the next step will trigger the expression of the IL-7 receptor, but for now, that's sort of the key. We sort of flip those switches of these two on and off to get the LMPP, which is still able to become either the myeloid line, including things like monocytes. I have to remember that picture from chapter 2. We had the myeloid line and the lymphoid line. So they can still become myeloid cells if they get the right signals, but they are primed to now move towards the lymphoid line. Okay. Once the IL-7 receptors and the recombination enzymes start turning on, we have the early lymphoid progenitor cell. Now it's made essentially the commitment away from the myeloid line towards the lymphoid line, but at this point could still become a T cell or a B cell. We're going to watch it go down the B cell, well, almost the B cell line. We go from the early lymphoid progenitor cell to the common lymphoid progenitor. We came across this guy in the last chapter as well for T cell development. Okay, so this can still become all of the lymphoid line, but now really pretty much no longer the myeloid line. So as you can see here, it's turning off c-KIT and SCA1 for the most part, really ratcheted up the IL-7 receptor. The recombination enzymes are primed to, at this point, either recombine the immunoglobulin genes if this continues to be on the B cell line, or the T cell receptor genes if it leaves at this point and goes into the thymus. Okay, so connect some of this early stuff with what we looked at in the last chapter for T cells. Okay, so if these common lymphoid progenitors leave to go to the thymus, they're going to commit to the T cell idea. If they stay in the bone marrow, which is what we're going to focus on here, then most of them are going to end up being B cells. Okay, so again, it's all about the specific microenvironment in terms of getting signals to turn on and off some of these various proteins that are triggering movement through this process. And so these are not just means of identifying the different stages, but they're presumably part of signal transduction pathways that are turning some of these other genes on and off that kind of keep nudging these cells away from becoming everything towards becoming something more specific. So that's really the key here. Does that kind of make sense? So I'm not necessarily all that keen on you memorizing all of these specific cell surface markers, but knowing that that's the general idea here. That as you get further and further away from that hematopoietic stem cell, it's making commitments towards a specific lineage, okay, because it's getting specific signals within different parts of the bone marrow in this case. Does that general idea make sense? That sort of theme makes sense? So we're going to pick up here. So in the last chapter, we followed those common lymphoid precursor cells out to the thymus, where they became T cells. Now we're going to stay in the bone marrow and watch them become B cells instead. Now why the difference? Well, because there's going to be a different set of cytokines being expressed and exposed to these common lymphoid precursor cells that are going to push it towards the B cell lineage, as opposed to all the cytokines and signals in the thymus that pulled those same cells towards the T cell line. Okay, so again, it's all about what specific signals these CLPs are getting within the specific environment that the cell finds itself. Okay. Because remember, if we took those and gave them different cocktails of signals, we got T cells or B cells. So again, it's all about those specific signals. Okay. Once again, we're going to look at some specific markers, again, as a means of how do we know that there are all these different steps. Most of it is based on the expression or non-expression, depending on the situation, of some specific cell surface markers and events that are happening inside the cell, mostly, in the case of B cells, the recombination of the immunoglobulin genes, which we've already studied. Okay? So we need to link this back to that chapter and remember how that's put together. And how the heavy chain goes first, right? We have the DJ event happen first, and then the V, then we have pre-B cell receptor, right, with the fake light chain, and then we recombine the light chain to get the real B cell receptor. Each of those steps kind of defines one of these stages in the development of the B cell. Okay, so I would say we want to focus more on that than all these different crazy cell surface markers, but there's a sort of a correlation between them. Okay. Just to point it out so you don't get confused, apparently there are two naming systems for these various stages. So the one in parentheses here, we're not going to worry about. We're going to do the other one that most people still use. So the ones in parentheses are the Hardy nomenclature. You can read about that in that specific experiment box if you want. We're going to go with pre-pro, early pro, late pro kind of thing, I think is better. Okay, so at this point, the first sort of specific B cell stage that those common lymphoid precursors are going to go through, if they stay in the bone marrow, is what's called the pre-pro B cell stage. And basically, this is just the starting place. Okay, so nothing has happened with the immunoglobulin genes yet, so therefore there's nothing to express on the surface, and they're pretty low on most of these cell surface markers, with the exception of this one called B220, which is the classic B cell marker. That's the B cell lineage marker. So remember I mentioned before this, these cells were lineage minus. Those common lymphoid precursor cells that stay in the bone marrow start expressing this B220, which is the B cell lineage marker. You'll notice once that's on, it stays on, because it's now made the commitment, I'm going to be a B cell now. Okay. So that's really the major thing that describes the pre-pro stage. It's sort of the first stage. It's made the commitment, I'm going to be a B cell, or I want to be a B cell. It starts expressing that B cell marker, and that's where things are going to kind of start. Okay. So we go from pre-pro to just pro, and people divide it into early and late, which is fine. So we'll start with the early pro B cell, which has started turning on some of these cell surface markers, but for us, I think the more important part is that the early pro B cell has started recombining its heavy chain. Okay, so it does the D to J portion of the heavy chain rearrangement. We go from early to late by finishing the heavy chain rearrangement. So the late pro B cell has rearranged its heavy chain completely. So it does the V part of the rearrangement. Now, this presumably is a little floopy because it has to finish trying one of them to figure out whether it worked or not, right? So presumably, if this one works, then we just continue down the line. But presumably, if that one doesn't work, and it has to try the second heavy chain allele, then it must have the ability to go backwards in that sense and try the second allele, which would then make an early one again, and then it would try, then it would do the do the V region and become a late pro again. So there's got to be a little floopiness in this because of the way allelic exclusion works and that kind of stuff. But not worrying about that too much. Let's just pretend it kind of all works the first time. Okay, so it does the DJ first, that's the early. Then it does the V recombination, and that's the late, what's called the late pro B cell stage. Let's, for argument's sake, assume that that was a productive rearrangement. Once it starts expressing that heavy chain along with the fake light chain, okay, remember how that works? It does the heavy chain first, and then creates that sort of two-part light chain, the V pre and the lambda 5, the fake light chain, and it starts expressing the pre-B cell receptor. Once you see the pre-B cell receptor on the surface, we go from the pro stage to the pre-stage. Okay, so these are pre-B cells that have successfully recombined their heavy chain and are expressing the pre-B cell receptor with that fake light chain, as I usually refer to it, or that surrogate, let's use the proper term, surrogate light chain. And remember, that is necessary to get a signal in order to recombine the light chain, right? So it does have its function, okay, so we need that in order to go to the next stage. So the larger pre-B cell is expressing the pre-B cell receptor, so that might be a way to remember that. And then I guess it shrinks a little bit in size, I don't know why, after it starts rearranging its light chain. Okay, so the difference between large pre and small pre is rearranging its real light chain. And of course, remember, we get 4 tries at that. Hopefully one of them will work. And let's assume one of them does. Once it is expressing the real antibody in the form of IgM, then we've completed this process, then we have the immature B cell. Okay, so this is the pre-selected B cell. Let's see. Oh, I was going to forget that. One thing I forgot up here. We have between the late pro and the large pre, we have this first checkpoint here. This is thematically similar to what we saw in the T cell receptor. Remember, it did the beta chain first. Yeah, did the beta chain first, the equivalent of the heavy chain, and then if that was productively rearranged, that B cell started proliferating. That was that beta selection. We have a similar thing here. If we get a productive rearrangement of that heavy chain, that's an important milestone in the life of this B cell, and that B cell gets to proliferate a little bit. Okay, so we have the equivalent thematically of that. So that's happening here as well. That's this first checkpoint. Which again probably does the same thing that it did for the T cell receptor. Now you have this little group of clones that all have the same heavy chain, but then will presumably all rearrange different light chains, and you'll get different specificities. So it's just a way to sort of expand the population of cells that have productively rearranged the heavy chain or the heavy chain equivalent if we were back in the T cell stuff. So that's this first checkpoint. Checks if you've got a productively rearranged heavy chain, you get to expand a little bit. And the second checkpoint is essentially as the cell starts to express that IgN on the surface. Okay, and the second checkpoint is really going to be selection, negative selection for all intents and purposes. Okay, so you want to have these, especially this particular series of steps at your fingertips. Okay, so what's happening in each of these steps, and again, focusing not really on these cell surface markers, but more on what's happening with the immunoglobulin genes and things like that, I think is the much more important piece of this table. Because these are nice in order to study and to separate all these different stages out from one another for experiments, and I think understanding what's really happening inside the cell is really more important for us. Okay. So it's really just adding on a title to the cell during each of these immunoglobulin steps, rearrangement steps. So if we really connect this back to that chapter, whatever chapter that was, we're just putting a name to the cell at each of the different steps of that process. Okay. Does that kind of make sense? Okay with that? Okay, so focus more on kind of the left-hand side of the table rather than don't memorize these things at all. I don't think that's really worth it. Okay, so now we've gotten the immunoglobulin stuff rearranged. We now are at the immature B cell stage, and I think we're pretty much ready to start selecting. This is just a sort of a detail slide, not that you need to memorize it, but it's just showing how sort of each of the steps that we've been talking about is going to be controlled by different aspects of a signal transduction pathway. Again, we can never ever get away from them, even though we're not studying them all that specifically or all that in detail. Each stage of these recombinations are controlled by different transcription factors that are part of signal transduction pathways. That's really all this is really showing us. Before we get to that, actually, there is no, surprisingly, there's not really a good picture for selection. But the general idea, I guess they don't draw one because it's relatively simple. It's a relatively simple concept here for B cell selection. As I mentioned earlier, all we really have to worry about here is negative selection for autoreactive B cells, right? Because for B cells, we never ever want them, theoretically, to latch on to self, right? That's a weird twist to the T cell story, right? Where we need the T cell receptor to be able to see self MHC, that's all. But that was a positive selection, and then if it saw real self, anything else that was self, we got rid of it. Here we just need to get rid of it. Okay, so there's really just negative selection for autoreactive cells. So there seems to be sort of three parts to this, in a sense, or three possible overall outcomes. The clear one is clonal deletion. If the antibody on the surface of the immature B cell attaches to a self-peptide, it is deleted, presumably by apoptosis. Right? That cell will go through apoptosis and be killed. So that makes sense. And we saw something similar in the T cell development. But I believe we also saw back in the T cell stuff that sometimes you'll get a second chance. So there is receptor editing as a possibility, so that self-reactive B cell, before presumably it's clonally deleted, might get a second chance. Okay, if it has more light chain alleles left, it can be given another chance to do that. So it could rearrange another one of its light chains, replace that with the one that was self-reactive, and hopefully the new combination is no longer self-reactive, and that cell gets sort of spared clonal deletion. So I think it makes a lot of sense. Why kill the cell if you can have give it another chance to tweak its receptor, and the receptor's good now, so why kill the cell outright? So those kind of go together, that they can be given another chance before they're outright deleted. And then the third possible outcome to selection is again where it's not killed outright, but it's, when we saw anergy for T cells as well, where it's down-regulated so that even though it's not outright killed, it can't be activated anymore. Now, exactly why you would want to not just kill the cell and just let it live but not be able to do anything, I don't know. I'm not sure anybody knows, but anergy does seem to be a possible outcome to selection as well. So it leaves the bone marrow, but can't be activated by antigen anymore, but it's still alive. So those are sort of the big three. Most likely, the first two are the major sort of processes where you're giving it another chance, but if none of the chances sort of pan out, then most of them are just going to be outright killed, which is probably the safest thing to do. Okay, so now I can move on this one. So that is all happening in the bone marrow. So after selection, those still considered immature B cells, but selected immature B cells, leave the bone marrow and then start populating various secondary lymphoid organs, like the spleen that's shown here, or lymph nodes, or wherever they go. The immature B cells don't have a very long half-life. They express high levels of FAS, which is one of the pro-apoptotic signals, because the idea here is we want to move this along to a mature B cell. Okay, so you don't want those immature ones around for very long, because at this point they're not ready to evoke an immune response. Okay, so if they never mature, we want that to happen. We want just to get rid of them fairly quickly. So we want to kind of move them along this process. So they enter secondary lymphoid tissue like the spleen, and as they kind of finish up their maturation process, two stages have been identified here that are called transitional B cells, and there are two stages, T1 and T2, and it's thought by basically everybody that they're only separate populations, but along the continuum. So T1 cells become mature into T2 cells, and T2 cells become mature B cells. Okay, so they're just a couple more steps along this maturation process. And how did they get identified? The same way people have identified all of these different stages by a variety of cell surface markers. So once the machine called the cell sorter was invented, all of this kind of stuff really blossomed, because before the invention of that, it's called a fluorescence activated cell sorter. Before the invention of that machine, you couldn't really do any of this stuff. So once that machine was invented, immunologists especially jumped on board and started identifying all these different stages and all these different subpopulations of cells that way. So again, it's a matter of expressing different cell surface markers where we can identify these different final stages along the maturation of these B cells. Okay, so we have T1s, T2s, and mature B cells based on these. So if we kind of look at it from that standpoint, from these T1s and T2s, it's really the T1 cells that are being selected. Okay, so here's the T1 that can undergo this negative selection if they bind to self-antigen. Okay, so these are dying through clonal deletion. So it's the T1s that are really going through selection. So we can kind of think about selection as sort of a two-step process. So part of it's happening in the bone marrow, another piece of it is happening here in these secondary tissues. Again, focused on the spleen, but this can also happen in the lymph nodes as well. So it's the T1s that are kind of going through negative selection. I don't like that they're using the term positive selection here. But what this really means is that those that survive the negative selection are positively, not really selected for, but sort of reinforced. So if you've gone through this sort of double negative selection process, both in the bone marrow and the secondary organ, then you should be okay. You're going to get positively selected to, kind of stay alive. That's what they mean here. I don't like that they're terming that positive selection, because it's not what we talked about as positive selection in the thymus. So I don't like that they're using the same word, but that's the general idea. Part of the slight difference between T1s and T2s are also that T2 cells will start expressing, will begin to start expressing also IgD. So that's part of the maturation process as well. Once the cell is producing or expressing both IgD and IgM, then we have a fully mature, ready to go, show me the antigen kind of B cell. So it's a kind of a long, laborious process to get from that hematopoietic stem cell through to this mature B cell who's ready to go, who's ready to say, I can look at antigen, be activated, and do what I'm supposed to do. We will get to that part of the story soon. Not in this chapter. They keep dividing the story, I think, in a few too many chapters, but what can I say? But we will eventually look to see exactly what B cells do. I know we kind of know generally what they do, but we'll look more specifically. Okay. So, here, now we'll go back to 2. Right, so the fully mature B cells are expressing IgM and IgD, okay, at relatively high levels. And becoming mature then also gives you the ability to sort of what's called recirculate. So mature B cells become mobile also in that sense. So T1s and T2s are sort of stuck in wherever they are, the spleen, the lymph node, wherever they are. Once they mature, we have both IgD and IgM, and they start to recirculate. Meaning, they start traversing the body, so they can stay in your spleen for a while, then enter circulation, move to a lymph node there, spend some time there, get back in the circulation, move to that lymph node there, spend some time there. They get to kind of circulate throughout the body, which makes some sense in terms of how all this, kind of where we're building towards. If the one B cell that you need to fight off infection X is there and stays there all the time, well then antigen X has to eventually work its way only there. Well, if it kind of moves around, and it's doing this relatively quickly, then you're going to kind of up the odds that the right antigen and the right T cell are going to get together. In mature T cells, we saw do the same thing. Okay, so it just kind of ups the odds that you're going to get antigen and the right B cell or the right T cell together more quickly. So recirculation is definitely an important characteristic for mature B cells. So just to put out some numbers, about 10 to 20 million B cells are produced every day, and only 1 to 3% will ever become mature B cells. So that just gives you a kind of an idea there. And we saw sort of similar numbers for T cells as well. The vast majority die, right, because they don't make T cell receptors that can hook up with MHC, right? That was the biggest death for them. Okay. Again, because of the random way that immunoglobulins are made, a lot of them are going to be against self-antigen, right? Because we don't know what they're going to be against before we make them. So we can only test for that afterwards. So a goodly number are going to be against self-peptides. We get rid of them through negative selection. So the vast majority don't become mature B cells. Let's see. Ah, let's do this quickly. Oh, sorry, and there are too many terms here. Mature B cells are also sometimes referred to as B2 cells. Although I don't think I've come across B1 cells, so I don't know what that's all about. Okay, but mature B cells are B2 B cells, sort of the same thing. So do that. And just one important one of these receptors to point out is this BAFF receptor, which is important for the continued survival into the mature B cell stage. You can see that that increases over time, so as it gets closer and closer to being a mature B cell, it needs more and more of this BAFF receptor to keep getting those survival signals. That's the primary thing there. And then technically, I don't think it's in the slide, but these energic T cells are sometimes referred to as T3 cells by some investigators. But that's where they stay. They never become mature T cells. All right, I think we'll stop here because I can't finish this in 5 minutes, but this will not take the entire next class. So we'll probably finish this up and maybe go into some of chapter 11 next time as well. We'll see how things go. Yeah, so that's what we'll do. Okay, so we'll stop here. We'll finish up B cell maturation, probably get